Hello everyone, hope you all are doing well. So today we are going to do topic 13 of paper 2, that is self incompatibility. Or in short we can write it SI. So it is the inability of plant with functional pollen to set seeds when self-pollinated. So here it is the inability of plant with functional pollen to set seeds when self-pollinated. In male sterility, what we saw there, it was the plant was not able to create or release functional pollen, but here plant is able to release functional pollen, but it is not able to set seeds when self-pollinated. So in short, it is the hindrance to self-fertilization. It is hindrance to self-fertilization. Now we will see the classification of self-incompatibility. Classification of SI. So the first one is based on flower morphology. So it is of two types: heteromorphic and homomorphic. In heteromorphic, we will see it is of two types: distally and tristally. In distally, we see that styles and stamens are of two types, that is short and long. Here styles and stamens are of two types. I hope it is visible. That is short and long, and it can be seen in, like for example, it can be seen in Primulaceae. And what happens in tristyles? Here the style and stamen are of three types, that is short, medium and long, and it is seen in Primulaceae. So it was about heteromorphic, and what hetero suggests here? Hetero, differ in morphology, that is you can see there is differ in the morphology of style and stamen. It is different in morphology, but in homo we have got same morphology, that is no difference in morphology. Let me rub this. In homo, what we will see, it is also here we have same morphology, but it is also of two types, that is sporophytic and gametophytic. In sporophytic SI, that is self incompatibility, is governed by genotype, but in gametophytic it is governed by genetic constitution. So it was the classification based on morphology. Now moving on. To number second, that is classification based on genes involved. So here we see that SI8, that is self incompatibility, is classified in three types, that is monoallelic, diallelic and polyallelic. In monoallelic, it is governed by single gene, that is one gene. In diallelic it is governed by two genes, and in polyallelic it is governed by several genes, more than two genes. Now, number third is based on site of expression. Based on site of expression. So it is also three types, that is stigmatic, stylar, and ovarian. In stigmatic, SI genes express on stigma, as the name suggests. In stylar, the self-incompatible genes express on style, and in ovarian, they express on ovary, as simple as the name suggests. Now, fourth type is based on pollen cytology. So it can be binucleate and trinucleate. So in binucleate, pollen grain can have two nucleus or two nuclei, and in trinucleate they can have three nuclei. So this was the classification of self-incompatibility. Now moving on, what are the measures to overcome self-incompatibility? How can we overcome SI? Measures to overcome SI, that is self-incompatibility. So the first measure is bud pollination. So what we will do here, we will pollinate immature buds with mature pollens. Pollinate immature buds with mature pollens. So it can be seen in Brassica, Nicotiana, Petunia, etc. Secondly, we can do delayed pollination. This is also can be seen in Brassica and Linium. We can do late season pollination. This can be also seen in Nicotiana, Petunia, Abutilon. Fourth, we can do irradiation. We can do irradiation with X-rays, as seen in Petunia. We can also do irradiation with gamma rays. Example, it can be seen in Nicotiana. We can also use high temperature, that is temperature over 30 to 60 degrees Celsius. It can be seen in Malus, Pyrus, Prunus, etc. We can also do multilation of style. So what is multilation? That is cutting of style. It can be seen in Brassica. We can do in vitro fertilization, that is IVF. Here we can also do in vitro fertilization, not only humans. IVF can be done in plants also. So what we do in vitro fertilization? We place pollen directly in the contact of ovules. Pollens directly to the ovules. It can be seen in Petunia. As we do in humans, we place the sperm directly to the near the ovary, directly at the ovary. So these are the measures to overcome self-incompatibility. Hope you understand this. Now moving on to next topic of this session, that is importance of self-incompatibility. So they are very important and used in hybrid seed production. They are very much used in hybrid seed production. Because SI provides a way of hybrid seed production without It provides a way of hybrid seed production without emasculation. We not without emasculation and without restoring. This is without restoring genetic or cytoplasmic male sterility. That is, this is genetic or cytoplasmic. In the previous lecture we have discussed male sterility in detail, you can see there. And it also permits combining of desirable genes, in a single genotype, from two or more different sources, more different sources through natural cross pollination, which is not possible through natural cross pollination, and this is not possible in self compatible species. It is only possible in self incompatible species. In case of pineapple, commercial clones are self incompatible. As a result, their fruit develop parthenocarpically and seedless. That is, what seedless fruiting is called? That is parthenocarpy. There is no rogging problem in self-incompatible line. There is no rogging problem here. Here the labor cost is less. And seed production is more. So these are the importance of self-incompatibility. Hope you understand it. Now moving on, the mechanism of self-incompatibility. So next topic of this session, and this is the last topic of this session, that is mechanism of self-incompatibility. Here we see that two events are considered to constitute the basis of considered as basis of self-incompatibility, that is stimulation of unlike genotypes stimulation of unlike genotypes and initiation of like genotypes. So these are the two events that are basic for SI, and thus two hypotheses have been proposed to explain mechanism. Two hypotheses are there. Is there. So the first hypothesis is complementary hypothesis. These are two hypotheses to determine the mechanism of SI or self-incompatibility. So this was given by Batman, not Batman, it is Batman. Batman is a superhero. He is also a superhero in agriculture, or you can say in botany. So this was given by Batman in 1952. So according to this hypothesis, SI results to self-incompatibility results due to absence of substances in the pistil or pollen which are very essential for pollen tube penetration. So Batman suggested that SI, that is self-incompatibility, results due to absence of substances in pistil and pollen. The substances that are required for pollen tube penetration are absent in pistil and pollen. That is why instead of having functional pollen grains, we are not able to do fertilization or pollination, what you want to say, or do fertilization. So this is what complementary hypothesis is. Now moving on to the second hypothesis, that is oppositional hypothesis. Oppositional hypothesis. This hypothesis states that interaction between like alleles let it be S1 S2 into S1 S2, so they are both same, that is like alleles, leads to production of inhibitor. Interaction of like alleles leads to production of inhibitor, which inhibits the growth of pollen tube in pistil. What this inhibitors though? It inhibits the growth of pollen tube in the pistil. Here the inhibitor can act in three ways. The first way is act in three ways. First, it may inhibit an enzyme or auxin necessary for pollen tube growth. It can inhibit enzyme or auxin which is necessary for pollen tube growth. And secondly, it may block pollen tube membrane. It can block pollen tube membrane. And thirdly, it can inhibit an enzyme necessary for penetration of pistil. It can inhibit an enzyme necessary for penetration of pistil. So this is how the inhibitor can act in these three ways, and this is what oppositional hypothesis. It suggests that interaction between like alleles, like genes, suppose these are two same genes, like genes, leads to production of inhibitor, and inhibitor inhibits like this way, like these three ways. It can first of all inhibit the enzyme, that is auxin, which can reduce pollen tube growth or stop pollen tube growth. It can block pollen tube membrane, and it can also inhibit an enzyme which is necessary for penetration of pistil. So I hope you understand this thing. So that is all for today. For any queries, do comment. If you like it, please press the like button. Do share and subscribe. Keep making notes, keep evaluating your notes. I always tell in every session that the notes you are going to make are your Brahmastra. Most of the question, almost every question from the topics I am going to teach you. So keep making notes. Thank you. That's all for today. Have a nice day.